Let's get started with today's webinar. First, we have Genique Lifesciences, a genomics solution provider dedicated to supporting labs, clinicians, and researchers with advanced molecular NGS technologies. We have someone better and more qualified to talk about Genique Lifesciences, CEO and co-founder, Mr. Abhishek Das himself. I welcome you to speak a few words about the company and our partnership with Genomize and Chromosome Labs.
I think Shrishti's put me on the spot with my qualifications, but I can comfortably say I am always the least qualified person at Genique, and hopefully it remains so. We continue to have more qualified people than me who are able to guide us, and more qualified customers and partners who we are partners with. Genique was founded in 2018, and our objective was to bring cutting-edge genomic solutions for labs and partners in India. And we're very proud to be partners with Chromosome Labs, who we've worked with for a long time, and also Genomize, who have a cutting-edge bioinformatics platform that works in a clear way. Without stealing both Dr. Heena and Dr. Tolga's thunder, I will allow Shrishti and Mandar to introduce them and speak about them. Our endeavor would be to continue to represent the best-in-class products with the best-in-class genetic labs in the country. We are a small team of about 14 people. Most of our presence currently is in the north of India. A smattering of people in the west and the south, and we look forward to having more such sessions with Chromosome Labs and Genomize.
Thank you, Abhishek. Now we have Genomize. It is a pleasure to collaborate with Genomize, a company based in Istanbul, Turkey, that specializes in AI-powered genomic data analysis and interpretation. Genomize has developed advanced bioinformatics solutions designed to help labs and researchers streamline variant interpretation, reduce analysis time, and generate clinically relevant insights from genomics data. Their platform integrates powerful algorithms with curated knowledge bases, helping clinicians and researchers navigate the complexity of germline and somatic variants. I request Dr. Mandar to introduce Dr. Tolga from Genomics.
It's my pleasure to introduce Dr. Tolga Aslan from Genomize. Dr. Tolga has completed his undergraduate and master's in molecular biology and genetics at Boğaziçi University. He later earned his PhD in molecular biology and genetics with a focus on bioinformatics from the same university, where his research focused on genetic analysis of rare inherited metabolic diseases in Turkish patients and the development of personalized gene therapy systems using primary tissue culture, animal models, and CRISPR-Cas9 technologies. Dr. Aslan currently serves as the head of customer success at Genomize, where he leads user engagement, supports the successful implementation of the SEQ platform of Genomize, and works closely with hospitals, laboratories, and clinicians worldwide to ensure effective use of Genomize's data analysis solutions. We are eager to hear you and your talk, Dr. Tolga.
Thank you.
The stage is yours.
Mandar and Shrishti made a very nice introduction for our company. But to reiterate, we are a clinical bioinformatics company, and we are specialized in preventive genetics, genomics, rare disease diagnosis, and personalized therapy for cancer patients. And our main product is called the SEQ platform, and it basically accepts any data type from all major next generation sequencing platform or variant calling tools, and we provide a comprehensive data analysis and accurate ACMG classification, which I will talk about in a few slides, and confident variant interpretation based on clinical information. So we are currently active in more than 30 countries, and we are happy to say that we have helped, so to speak, in analysis of more than 400,000 patients from more than 450 different hospitals and research laboratories around these countries. So first of all, I want to focus on or touch up on a few things, challenges, what we call challenges in clinical bioinformatics, right? So, it's a multi-step process. And of course, the first step after, of course, the data generation is variant calling. So the main challenges here are accuracy standardization and medically relevant challenging genes. So, the list actually is much longer than that, but these are the major items. So to solve that, we have to use, a proven algorithm to call the variants, and we have to have 100% repeatable pipeline versions. This is usually overlooked, but a good bioinformatics tool should always give you the same result when you start the analysis with the same data. No matter how old it is, like if you reanalyze your sample from 5 years ago, you should always get the same results using the same pipeline, yes? And also to overcome the medically challenging genes, you have to use some special types of variant calling. So after we call the variants, let's say, we are working with a whole exome in which you will have typically hundreds of thousands of variants, or if you do whole genome, you will have millions of different variants. And then the next challenge is to classify the variants. And of course, ACMG classification is a center stone of our analysis, everybody's analysis throughout the world. But there are some challenges in ACMG classification, which I will again talk about in a few slides. And the next thing is there are numerous data sources. Well, it can overwhelm the analysts with the plethora of different data, and sometimes different data sources provide the same class of information, but they provide different answers to your questions. So you have to be very selective in selecting your data sources. And also another challenge is the abundance of transcript isoforms. Usually in clinical genomics, the canonical transcript is usually analyzed, processed, or evaluated, and then the alternative isoforms are just disregarded, yeah? And the issue with this is, and this is also suggested by the guidelines, we have to process and analyze and evaluate every single transcript which can be clinically relevant. And for that, of course, we have various solutions in the Sync platform. And after you classify the variants, of course, the next challenge is the shortlisting, because even after you classify based on, let's say, ACMG classification, you will still have maybe thousands of variants in a typical whole exome sample. So the solution for that is, first of all, either you as a person or the software should identify the clinically relevant genes and offer you some solutions in resolving the variants of unknown significance, and also be able to create a database for you. And the specific reason for that is there are many regional polymorphisms which are not represented in publicly available data sources. And that information becomes very, very important in making your final decisions. For example, in my own genome, I carry a variant which has zero occurrence in NOMAD database, but the frequency of that specific variant is around 20% among the Turkish population. And it's in a very important gene for hearing loss. And because it is represented so much in my own community, I'm not worried about that. Because hopefully, it's not going to have any effect, it will not cause anything. So, and of course, there are some other solutions, which I will talk about in a second. And the last step, actually, I started to talk about that already, is data aggregation and sharing. Because, as I mentioned, there are many underrepresented populations in publicly available databases, and your local, even your own cohort, of your own clinic and from your own country or geographic region becomes very, very important. And also, as you know, science is based on sharing, right? We in the research labs, we do some research and then publish our results, so we gain knowledge as the humankind. So in that regard, as a small part, actually, data sharing is quite important in genomic analysis as well, because actually what I have given you as an example from my own genome, from my variant, is a very good example. If we didn't share that information among the Turkish community, then I would be kind of worried, I believe, and going from clinic to clinic, if I should be worried and are there treatments, etc. But since there is anonymous data sharing within the SIG platform, again, I have enough information to just eliminate that variant from my mind, right? So I will touch upon a few things, going into tiny bit of detail. So as I mentioned at the beginning, the variant detection is very, very important. This is the maybe one of the most critical steps. And for that, there are various, of course, variant callers. And according to FDA Truth Challenge, there are 3 variant callers that are ahead of others. Namely Deep Variant, Centurion, and Dragon. Centurion and Dragon are already supported in the SIG platform, and Deep Variant, we are working on it, and we are performing the benchmarks as of today. Also related to this, of course, SNVs are not the only variant type, and detection of copy number variation is also very critical in today's world. And of course, as a scientific company, we perform the benchmarks, and these are our results with very high sensitivity and specificity ratios. But again, as a scientific company, we try to think about what will happen in actual clinical setting. So as you can see from this graph, there are two main factors which affect the sensitivity of copy number variation detection. And these are, first of all, the sequencing depth and the cohort size. Of course, in our benchmark, we use the 600x depth samples, and which is not always possible to reach that depth in a clinical setting. So if you reduce the depth to 60x or 150x, as you can see, the sensitivity drops. But the thing is, the specificity, which means, are the copy number variations correct, basically, remains close to 100%. The factor that is affected is the sensitivity, which is the ability of the algorithm to detect the copy number variations. So moving on, I will talk a little bit about ACMG classification. Of course, as I mentioned, this is the center stone of our variant evaluation throughout the world by everybody, basically. And the softwares need to be very transparent about what they are doing in the background. So this is what we try to do. If there's a decision tree, for example, we try to show you exactly which steps it went through. But what I want to focus on is the performance, of course. So for that purpose, we performed a benchmark last year, if I'm not mistaken, with 6,200 variants directly taken from the Klingon manually created variant dataset. And actually, we have evaluated the data like 2 weeks ago with the updated list, which included a couple of more thousand variants, and the results were practically the same. So this data set, again, contained a little more than 6,000 variants from more than 150 genes and different types and different diseases. So the results are somewhat striking, I have to say. So on your screen, you see the Sankey plots of the let me use a laser pointer here, of the classifications of the variants by Klingon expert groups, and their classification of the same variants in the Seq platform and one of our competitors. And the next slide will show you the other competitor. By the way, these competitors are very well known softwares and being used very frequently throughout the world. So the first thing I want to take your attention to is our competitors consistently overclassify or misclassify the VUS variants of unknown significance. So as you can see, about a third of the VUS are classified as likely pathogenic, and almost all of the likely pathogenic variants are classified as pathogenic by this competitor one. We are not perfect either, but unfortunately this is by design of the current guideline, which will be changed quite soon in like 6 months or 7 months. We will have the new version. Hopefully it will solve these issues, but with the current guideline nobody's perfect. But our concordance with the Klingon manual curation is much greater than competitor one. And this trend is also correct for the competitor 2, but competitor 2 tends to distribute the VUS variants to likely benign, likely pathogenic, and pathogenic. So I believe I don't have to stress out how important it is not to report a variant of unknown significance as pathogenic or likely pathogenic. So moving forward, as I mentioned in my first slides, after the classification of the variants, the shortlisting is the next challenge. So for that purpose, we have an integrated sorting algorithm, additional classification algorithm within the Seq platform, and it automatically classifies your variants. And this is some information about the real-world data set that we have used for benchmark. And we use 215 diagnosed whole exome sequencing samples, and our algorithm basically classifies each and every variant, no matter how many variants you have in your sample, into 8 categories. So these 8 categories are, of course, I'm not going to go into details right now, but they basically sort the variants or classify the variants based on their clinical significance. So based on that significance, you see some top-X analysis. In other words, if a case is diagnosed, in which order the variant is reported within the software, right? Pretty basic. So with our algorithm, if you provide disease and phenotypes, the specific information about your case, around 50% of the time, the causative variant will be in the first place. And 82% of the time, it will be in the top 5, and the trend goes on. And to compare it with the conventional way, applying the filters yourself and sorting by ACMG, etc., the first place ratio is 14%, and top 5 is 43%. So there's a significant improvement in. I have to say, we don't do anything else, right? This classification does not affect anything else, just the sorting. But the important thing here is we save time when doing our analysis, because I don't have to apply filters. It will be right there. Few scrolls, I'm done. So let me just skip that. And a quick information about data sharing. In the Seq platform, a few databases are created real-time for you. And one of which, of course, is your within center frequencies, and the other is the community frequency. So I don't want to get into details, but how we process this information is the scientifically correct way to calculate frequency information. It's exactly the same how Nomad, for example, processes their data. And we don't simply show incidences to you like most of our competitors do. The way we do it is much more expensive and much more resource hungry. But since this is the correct way, we do it this way. So let me skip these slides. And there is a new addition to our database functionalities, which is the CNV database. So if you start, when you run our CNV pipeline, we create again a real-time database for you, so you will know exactly in which patients of your cohort or your previous patients you have seen this copy number variation, what is the overlap, what is the copy number in that patient, and what is the phenotype and diseases that we have submitted for that patient. And this will allow for quick dismissals or filtering out of the copy number variation or quick reporting. Because as you know, unfortunately, we as a scientific community do not have as rich resources, I'm talking about databases and knowledge bases, as we do for small variants. So this is a very nice addition in our CMV pipeline. So of course, the SIG platform supports somatic analysis as well. And in this case, we use a specialized database, namely Genomic and CKB, to assign tier information to variants. And here you see a small screenshot of our variant interface. But the thing is, in today's world, variant and tier associations alone is not sufficient. So, of course, you need the clinical trial information, but more importantly, day by day, the treatment options that requires more than one evidence, which can be a variant or a biomarker or anything else, is increasing. So right now it is around 10% of the total evidences, but day by day it increases. So therefore we also show you, which I unfortunately omitted to include that screenshot in the presentation, but we also show you the evidence point of view. So treatment, this treatment requires these variants, so to speak, and your patient has these, so you can prescribe or treat your patient with the solution. Let me skip that slide, but this is basically another database functionality for tumor mutation burden and MSI. And in summary, there are significant discrepancies in ACMG classification between different algorithms, different softwares, but our algorithm has higher F1 scores, and our variant prioritization algorithm is 97.3% accurate in identifying the causative variant in prioritized tiers, and technology-specific phenotype, genotype data aggregation for both small variants and copy number variants, a distinguishable feature. And Genomize-seq is usable for both germline and somatic pipelines in conjunction with Genomic and CKB cancer mutation database. So this is the end of my presentation. I believe I have like 5 minutes or so, yes? So please correct me if I'm wrong. I don't want to,
Yes,
Thank
yes.
All right. So let me very, very briefly show you the product. So our product, the SIG platform, is a web-based, cloud-based, software. Of course, we have several servers, but we have the option to install on-prem. In other words, if you have a capable server, we can install the software in your server, so it will be completely isolated from the internet, so to speak. So I will be very brief, but this is a sample, this is an actual clinical sample. And of course, we provide you with a lot of quality control information, like the sex prediction, the number of free, heterozygous, homozygous ratios, transition, transversion, etc., as well as runs of homozygosity information. And also you can perform CNV and multi-sample analysis. And all of the things that I have talked about in my slides just basically comes up to this, right? So this is the small variance tab, and here you see basically any information that you will require in analyzing the data. And in this sample, for example, this variant is the causative variant. By the way, this is not a cherry-picked sample. This is the same sample that we have been using for 8 or 9 years. Because we are not a clinical laboratory, it is very hard for us to get consent. So for this one, we have consent. So we use this one over and over again. So this variant coming at the top is, again, simply by, I don't want to say coincidence, but the power of our algorithm. So here you see many information. So here is the database, the variant database I have talked about, and of course we have integrated HPO. So you will see the overlapping HPOs of your samples of the gene with your patient, and we have many options to filter out, sort, and what have you with your variants. And we have, of course, some practical things. You can assign your samples to anybody in your laboratory, and you can assign. Let me assign it to Bade. Of course, normally she would do that, but you mark this as diagnosed, and your team will know, okay, this patient is diagnosed, etc. So here I want to mention a few other things. So yeah, of course, as I mentioned, you can perform copy number variation analysis, and we use GATK CNV pipeline as well as DELI to call split reads from the sample. And again, this is the database that I have talked about. So you can see how many samples and their copy number variations overlap with your own sample, what are their phenotypes and diseases, etc. And you can play around, so to speak. So currently it's limited to 50% overlap, but I can drop it down to 20. Let's see if things change. It didn't, but because my demo account has very limited number of samples. And again, I have very limited time, but we have a lot of information that we provide to you. Lastly, I want to very briefly touch up on long reads. So we have support for long reads, both PacBio and ONT. So if in the future you have some samples, you can always get in contact with Genique, and we can arrange some demo, etc. So we have support for structural variants, of course, as well as targeted callers based on the technology that you are using. So I believe, without going over my allocated time, this is it.
There we will be understanding a user case, and so both the users are from Chromosome Labs. And firstly, I would like to introduce the Chromosome Labs, and then we'll move to the speakers. So we are delighted to collaborate with Chromosome Labs, a leading genomics lab based in India and an avid user for Genomize solutions. They have been actively involved in clinical genomics testing and interpretation, supporting clinicians and researchers with high quality genomic diagnostics and reporting. Their expertise is in translating genomics data into clinically actionable insights, which makes them an invaluable partner in conversations around germline variant interpretation and reporting strategies. Their experience from real world cases will add a very practical dimension to the today's discussion. I request Dr. Mandar to introduce the speakers from Chromosome Labs.
Thank you, Srishti. Yes, it's my pleasure to introduce Dr. Heena Satam. She's a senior scientist at Chromosome Labs. Dr. Heena, I've known her for a long time. She's a PhD in reproductive biology from an NIRH, an ICMR institute, and she has witnessed the changing trends of molecular diagnostics from simple and point PCR diagnostics to the current trend of high throughput genomics. She has been working in the field of clinical genomics since last 3 years, handling analysis, interpretation, and reporting of the whole exome sequencing data, along with bioinformatics team at Chromosome Labs Private Limited, Mumbai. and she has gained a sound knowledge of interpretation guidelines from ACMG AMP during her career path in clinical genomics. Her contributions are instrumental in ensuring accurate and clinically actionable report that impact patient care. Her passion for genomics combines with her technical knowledge makes her the valuable asset to the company. High quality variant curation workflows based on the clinical details, identifying clinically relevant variants and interpreting them in accordance to AMP ACMG guidelines using Genomize as one of the tertiary analysis tools are the key features of success in whole exome sequencing reporting at Chromosome Labs by Dr. Heena and her team.
DNA is the basis of all the phenotypes or the diseases, and it connects the dots between the precision medicine and the diseases. So as a central dogma, theory says that DNA is the basis of molecular biology. Now with the omics coming into the as a evolutionary biology, DNA is still the basis of the genotype-phenotype driven pathway. So when we know that basics of the phenotype is hidden into person's DNA, it can be an eye color, height, obesity, anything. So this DNA is nothing but it's as good as a DNA 3 billion letter book, which decides how your body should develop and how your physical characteristics should be and how it should function. So this branch of genomics, it deals with the changes that occur at the DNA level in this 3 billion letter book, and it associates these variations are then associated with the phenotypes, clinical phenotypes. Now, Srishti, next slide, please. Hello. Next slide, please. Yeah. So what are these genetic variations? So in humans, previous slide, previous to that. Is it possible if I control the slides, Mandar? Or
We'll see if we can
okay.
get you.
So it doesn't matter. So as we know that in the human genome, which is 3 billion sequence, we have 99% homology, which is shared between the two individuals, but there is only 1% variation, which makes us different. Now this difference is can be a polymorphism and can be a mutation. So these normal phenotypes, which are determined by the polymorphisms, and the disease phenotypes, which are lethal changes which may occur in the DNA at in this 1% of sequence, makes the person vulnerable to some diseases and these variations are different types of substitutions, which are missense, nonsense, or silence mutations, could be insertions, deletions, duplications, and translocations. So the way we read the book, where we have, when there are some errors happen in the book, like some paragraphs are missing or some letters are missing, there are some pages which are missing. Similarly, in human DNA, we have these types of variations, which are called as the genetic variations, and the genetic analysis looks into these variations through sequencing. Now, there are different methods to understand these variations, because these variations can be as small as one base pair change to a large chromosomal rearrangements, such as large deletion, duplications, translocations, inversions, insertions. So when we want to understand which technique to use, which testing method is to use, we first need to understand which type of variant we want to look into. Next slide, please. So in this, the genetic testing, it evolves from the microscopic level changes, which are captured either through chromosomal, through karyotyping or spectral karyotyping, to thousands or hundreds of bases of submicroscopic changes, which can be captured through FISH or chromosomal microarrays, and to the smallest fine level changes, which are at DNA level, which can be from one base pair to up to tens or 50s, hundreds of base pairs through Sanger sequencing or NGS. So when we want to understand, since we know that these changes are genetic changes, technologies have evolved now. With this evolution of the genetic testing methods, we understand the basis of the diseases, like why certain people respond to certain therapies, why there is some cancer which is familial in families, or certain cancers are prevalent in families. Why some children are autistically developed or they are intellectually disabled. So all these phenotypes which are imparted to the human being is because of the changes in the genetic sequence, which is part of the 1% variation. So next slide, please. So now what are these variations? There are different types of variations. This is a very basic information which is required when we are trying to annotate the variants and trying to interpret the variants. So at the variation levels, there are two types. One are the genetic gene levels or the nucleotide levels, and the others are the chromosome levels, or they are called as large structural variations. So at the DNA level or the nucleotide level, there are SNVs or they're also called as substitutions, which can be missense. Changes in this one nucleotide change can lead to the change in the amino acid code, or it can lead to the change in the amino acid, which is a stop codon, which makes the protein, which makes the amino acid nonsense or stop amino acid, or a small or a silent change, or it is a synonymous mutation. Similarly, small insertion deletions, which are from one base pair to up to 50 base pairs, which can lead to a frame shift or a non-frame shift or in-frame deletions or deletions. Then there are splice variants, which are located at the splice acceptor or donor sites. Now, the splice acceptor donor sites are very important because when we are looking at when we are applying some algorithms or some tools or some criteria when we are interpreting the variant, the understanding of the SNV changes, if they are affecting the splice sites, makes the variant pathogenic rather than simply moderate or low evidence. It gives a high evidence to the variant. Then in the chromosome changes, or which are structural variants, we have large deletion duplications, which can be more than 1 kbs, and there are more than that large insertion duplications or large chromosomal rearrangements or translocations, which are still higher. As I mentioned, they are microscopic changes and needs either whole genome sequencing or karyotyping. So the CNV part will be covered by Dr. Kumar in the next session. So in this session, we will be concentrating only on the nucleotide changes, which lead to the variants and the interpretation. Next slide, please. So now when we know what are the types of variations, we also need to understand how they are labeled. Because it happens that one lab may label a variant only using the codon number, and it uses the nucleotide base along with the codon number, whereas other lab may use just a cDNA position to indicate the variation, and the third lab may use a nucleotide position, nucleotide change only at the codon position, and does not indicate any of the genomic positions. So this differs in the interpretation, this may differ into the interpretation of the variant, or whenever somebody wants to search for the variant, whether this is already reported variant or any information if they want to retrieve. So there should be a consensus unique labeling system which was required, and that is called as the HGVS nomenclature, which was developed by Hugo, and it sets some rules or patterns when you are doing a variant labeling. So as this example is shown, first is the reference ID which is given, which reference ID can either be a genomic reference ID or a genomic sequence, or it can be a transcript sequence, or it can be a protein sequence, reference protein sequence. This is indicated by NC or by NM or NP, followed by the ID or the reference ID. Next to that, there is a colon, which is a separator, which is followed by the actual position of the variation. And if we are referring the genomic ID, then G dot is added. If we are referring the transcript ID or the mRNA sequence, then C dot is added. If we are referring the protein, then P dot is referred. Post that, different variants are added based on the type of variation. The symbols are added to it, like substitution is indicated by this symbol. Then silent mutations, del duplications are indicated by del or dup as the prefix. Insertions and the frame shift by plus or minus signal with the asterisk into it, with the FS. and so on and so forth. So these unique labeling system help us in understand when we are annotating the data, annotating the variant, which we gain through a raw VCF file. This unique labeling system helps us in annotating the other different functional and biological parameters to this variant. Next slide. So now we have, with the evolution of the genomic sequencing technologies, person comes to you, a patient either with the known genetic disease or with a suspected genetic disease or phenotypes. He comes to you, we do the sequencing. We do a high throughput sequencing, we align the sequence with the reference genome, and then we get millions of variants from the raw VCF data. Now, what should we do with these variants? Which one should is important for my patient, and which one should I choose to report it? This is where the annotation and interpretation of the variant comes in. Next slide, please. So annotation is nothing but adding the biological and functional context to or evidence to the variant with the help of computational tools, algorithms, and pipelines. So now this is a picture which is exported from literature, where you see that VCF file, each VCF file, which is a raw VCF file, which is unannotated, is submitted to the different pipelines, where the annotated databases are integrated. It goes through different it passes through different population databases, disease databases, and then it assigns it assigns a unique identification to this variant, which is called as the annotated variant. So we get the annotated VCF file. Next slide, please. So we get the annotated VCF file. You can see the table below. This is a small example of annotated VCF file, where we get the gene name is assigned to the variant, because in the raw VCF file we just get the chromosome, the position, and the reference and the alternate allele. But after passing it to the annotated pipeline, we get the complete detail to that. We give complete detail to that variant. Now, using this variant raw VCF file, this is our simple workflow, how the annotation and the interpretation workflow works. So these are submitted to the secondary analysis, where variants are annotated. We use the Genomize SEQ platform for secondary analysis for some of our projects, and some of the projects we use the tertiary analysis of Genomize. These annotated VCFs are then interpreted using tertiary analysis of SEQ platform. Then the variants are assigned a clinical significance. Then these variants are then we understand that these variants are the cause of the phenotype in your patient, and then the report is prepared accordingly. So we'll come to that exactly. We'll go through some case studies where how do we assign the phenotype to the and we ascertain the variant which is classified through Genomize. How do we ascertain to be added into the report? So annotation is nothing but it gives you a complete portrait of your variant with reference to the literature which is available. So once you have the annotated VCF, it will tell you about what is the variant type, annotated VCF, the variant to the variant. We will get the information about type of the variant it is, what is the molecular consequence of that variant, which gene it falls into, if there is any coding or non-coding region into the, if it falls into the coding or non-coding region, what are the amino acid changes, and if this variant this is now into the form of annotated variant. So it is easier for the annotated pipeline to search the variant into the literature, and it will fetch the information whether this has already been reported in the literature and with what clinical significance. Next slide. So once we have the annotated file ready. So these are the different databases which are used for annotation, population databases, then genome population databases. Next slide. These are very well-known population databases, DB, SNPs, and Nomad. Then disease databases, ClinVar, OMIM, HGMD. Next, then sequence databases. So the annotation uses all these databases to assign the different parameters to your variant, which is obtained through sequencing. Next slide. Okay. Also, apart from this, it uses predictive algorithms to assign the prediction of to the variant, whether if it is a SNV, whether it is a small insertion deletion, how it is affecting the protein function and protein structure, and whether it has been conserved evolutionary. So all this information is very helpful when you are doing the annotation, and all these are integrated into the softwares, the secondary and tertiary analysis softwares. So Genomize uses some of these features to for the annotation and variant classification. Next slide, please. Next slide. So the what earlier slide had the missense, the predictive tools for the missense variants, whereas there are some tools which are used for splice site variants also. Now, once we have the annotated VCF, annotated variant, some labs, here's just a small example which is given. Some lab may call it as a pathogenic variant, other lab may call it as a benign variant. So as per the evidences which are gathered by lab A, they may not find it as very important to be and may get evidences which point out it to be a benign variant, whereas the other lab gets the evidences which it points it out to be pathogenic variant. So there is no consensus guideline to assign the classification or the pathogenicity to the variant. So with this, which was creating confusion into patients as well as into clinicians, what to take, what message should we take, and what actionability we should do. So with that, the SEMG and AMP came together in 2013 and they form a guideline which is called as a very well known guideline called as SEMG AMP guidelines. So in 2015, and now this has been used by worldwide for classification of the variant. Next slide. So this guideline it defines it uses total 28 attributes or 28 criteria to define the variant, and it classifies the variant into 5-tier categories, like either pathogenic, likely pathogenic, variant of uncertain significance, likely benign or benign. Now these categories, 28 attributes, they are grouped under pathogenic categories with 16 criteria and 12 criteria for the benign call. So if the variant does not fall into either of the categories, it is classified as a variant of uncertain significance. Or if there are evidences which are assigned to this variant which are contradictory, if some evidence gives it a pathogenic classification, whereas the other evidence gives it a benign classification, in that case also the variant is classified as a variant of uncertain significance. So when we are assigning when we are interpreting the variant, the only symptoms, patient symptoms, family history, the suspected disease is not enough to interpret the variant. We need to have a consensus guideline to assign it to the variant, which is followed by the SCMG guidelines for the Mendelian single gene disorders. Next slide. So now these are these 28 attributes, as I mentioned, they are captured through different 8 categories or disease data sets, which when any algorithm or any software classifies the variant, it passes the variant through these 8 categories: population data sets, computational data or in silico prediction, which has the functional impact predictions, then functional data or the experimental evidences which are available in the literature, the segregation data, if available, or if there is information available with the clinician, with the family studies, then de novo data, then where we need a parental testing, whether parental testing has been done or not. Accordingly, the criteria is given to the variant. Then allelic data sets, whether what is the allele type, whether it is heterozygous, homozygous, substitution, or what kind of allele it is. Then disease association studies with referring to different literature, disease databases, and variant type and location. So based on all these categories, the variant has been attributed with either of the some of these 28 attributes. Next slide. This is just list of 16 pathogenic criteria. What is the definition of each of them? For example, I'll not go through all of them. This is available on in this reference of 2015 by Richards et al. So, for example, if the variant has a it gives a loss of function, then the variant is classified as has given a criteria of PVS1. So this can be through a computational prediction tools if it classifies it as a loss of function variant. Similarly, for PS1 criteria, which is a strong evidence for evidence of pathogenicity, PVS is a pathogenic very strong evidences. Similarly, PM is the pathogenic moderate evidences. So based on the combination of evidences, SCMG has set up some rules of combination of criterias which help us in classifying the variants. So all these definitions I will not go through, but when I'm going to the case studies, I will take you through individual case to case basis evidences. So next slide, please. So these are the rules for combining the criteria to classify the sequence variants. So for very strong variant is, I'll explain one or two cases. For a very strong, for a pathogen variant to be classified, it should have one strong variant, and apart from that, it should have either one or more PS1 variant, PS variants, or more than two moderate variants, or one moderate and one supporting evidence, or it should have two or more supporting evidences. So PVS1 is very important. Apart from that, you need either of these ABCD categories. For the second, or if you have more than two or more than two strong evidences, then also it can be classified as pathogenic. Or third, if you have one strong evidence and either of these ABC categories. So this is how the classification criteria is used when you are combining the criteria and want to classify the evidence after giving the attributes to each of the variant based on the 8 categories. Next slide, please. This is for the likely pathogenic. Similarly, you need to understand this, how this likely pathogenic criteria are given. Whereas you have one PVS1 and one moderate one, it can be called as likely pathogenic. If you have one strong and one or two moderate, it can be called as likely pathogenic, so on and so forth. So for VUS, as I mentioned, if there is a contradictory or contradictory attributes given, pathogenic as well as the benign, then the variant is called as variant of uncertain significance. So now how does this help the clinicians in taking the clinical decision? Like if you report a variant as pathogenic, then it has the clinical effect. It has direct correlation with the disease phenotype, and it is actionable variant. The clinician can take the action on it. Of course, the genetic counseling is the core of all this genomic testing and genomic analysis and reports. Then if it is a likely pathogenic variant, again, it is actionable variant and it can be taken for clinical decision making. Fourth is the variant of uncertain significance, which is also reported. Some labs report based on the requirement of the clinicians. At Chromosome Labs, we report the VUSs because it may have implications in future, or if the clinician wants to get back to us, if he's aware of what VUSs are called for his patient, and in future, if he wants to retrieve the data, he can come back to us and we can do the reanalysis if there is any if there is any upgradation or has been happened to this variant for the clinical actionability. Likely benign and benign variants are never reported, but they are at the part of the data set of the laboratory. The report, what does a clinical whole exome report include? So it reports the phenotypes which are used for the analysis, apart from the clinical history, which is a part of the demographic of the report. The Genomize or any other software puts the phenotypes for the analysis, which is called as a phenotype-driven analysis. Then there is a final result interpretation column where primary variants are reported. Primary variants are the phenotype-driven variants, and that explain the patient's phenotype. These are the actionable variants, either pathogenic or likely pathogenic variants. And there are non-actionable VUSs are also reported as the primary variants. The secondary actionable findings, as everybody is aware, secondary ICMG has has given a list of 83 unique genes which have to be reported if they are found to be pathogenic or non-pathogenic or likely pathogenic in version 3.3. Which covers 4 unique phenotype categories like cancer, cardiac diseases, metabolic diseases, and miscellaneous conditions. And these are conditions which have to be reported in the patient's report and genetic followed by the genetic counseling. Then there is additional finding on non-actionable incidental findings. These are again as per the lab policy. If they are pathogenic or likely pathogenic, but they are non-actionable at the time of reporting. They could be non-actionable at the time of reporting. But why do we report this? This may be helpful in differential diagnosis with the overlapping symptoms. If the clinician wants to understand if there are some overlapping symptoms and his patient can fit into the incidental finding, then they may have carrier implications for future generations. They may have implications in late onset of diseases such as ataxia or late onset dystrophies. There may be a risk modifiers for certain conditions such as cardiac risk, Alzheimer's, Parkinson's diseases. So that is the reason the incidental findings which are pathogenic or likely pathogenic but may not be directly related to the present phenotype are reported. Then variant gene details are also captured, which are nothing but the annotated details of the variant, the chromosome position, chromosome number, and what are the OMIM phenotypes which the gene has been assigned with, what is the depth of the variant. These details are captured into the variant gene table, and the gene which also the gene which from where this variant belongs to, the details of that gene is also included in the report to know the role of this gene into other phenotypes as well. The QC matrix is very important part of the report. Next slide, please. So this is how that variant table which is reported for the primary variants as well as for the secondary or the additional finding variants. So when we are reporting the variant, once the variant is now classified, how do we select that the variant should be reported, should be added into the report or not? So it depends on the quality matrix of the variant, whether it fulfills the quality parameters of the criteria, whether this is association of the gene is with the patient's phenotype, this zygosity of the variant. Suppose the variant is the gene has the autosomal dominant inheritance, recessive inheritance, and I am finding a variant which is heterozygous, then I may be calling it as a carrier variant. So that is what is the zygosity of that variant present for the condition. Then this is inheritance and the relevance. This is what I meant. What is the disease inheritance in the OMIM and relevance with the patient zygosity and inheritance pattern in the family, if the family history is available. The phenotype segregation data, if available in the family. Population frequency data, whether it is a de novo variant or if there is any parental testing which has happened into this family. So these are again the attributes from the SCMG attributes which are already assigned through the software. So we can take a relook at these attributes which are software driven, and then we can decide on to reporting of the variant based on the SCMG classification as well as the individual patients' phenotypes and the family history. Next slide, please. So now coming to the case study, this is is it visible? I think it's very small. Is it visible? A case,
Yes,
this is
it's
a
visible.
26-year male which was born to a non-consanguineous marriage. It's young onset of rigidity, asymmetrical tremors, bradykinesis, under-responsive to levopoda and Parkinson's disease was suspected. So when this analysis was done, these are the analysis phenotypes which were added into the software during analysis for prioritization. These are the quality matrix which was added into the report. So on the prioritized variants based on phenotypes, when we did the filtering of the variants, the prioritized variants which were popped up in the Genomize, as Dr. Tolga mentioned, based on the advanced algorithms which are added into the Genomize, we get AFGL AFG3L2 gene with the variant nomenclature as so and so. The coverage under zygosity and the consequences is 136x coverage, which is very good coverage. The zygosity is heterozygous and it's a stop gain variant. Okay. So now this disease with this gene, when we looked at the OMIM, this gene is for autosomal recessive SCA, spinocerebellar ataxia type 8. So definitely this goes with the phenotypes which have been submitted. The ACMG criteria which was assigned to this variant was PVS1, which is loss of function variant. Then it has PM2, absent in population databases. So one variant, one PVS plus one moderate, one very strong evidence plus one moderate classifies it as a likely pathogenic variant, and hence this variant was reported. In this patient, we also had one SPT1C1, SPTLC1 gene, where the coverage was good, it was heterozygous, and it is a splice donor variant. This disease is linked to juvenile ALS 27. There's autosomal, which is autosomal dominant, then autosomal dominant hereditary sensory and autonomic neuropathy, which is again autosomal dominant. Though this is a VUS variant, the pathogenicity which was assigned was PM2. So it is only one moderate. There is no other criteria which was attribute which is given to this, and hence it falls into the criteria of VUS. And we have reported this because this is autosomal dominant gene variant, and it matches with the patient's phenotypes. Though we have one likely pathogenic variant, we have reported this as a VUS into this patient. There's one more VUS which was added to this, which is TRPM7, which is again a synonymous synonymous variant, but this is a splice region variant, and it has attributed to PM2 as the variant classification. Which is again for the autosomal dominant in the OMIM for ALS, Parkinsonism, dementia complex. And so based on the type of variant, the detailed phenotypes. So whenever we report, we put a remark of the genetic clinical correlation. So if some phenotypes which are maybe missed by the clinicians and the variants which are reported, based on that, the clinician can take a call whether to take a action on this variant or not. So whether with the likely pathogenic and pathogenic variant, definitely he will take the action because the variant matches with the clinical phenotypes. But VUS and are reported because he is a young patient, and if there are other phenotypes which may develop, which may match with the juvenile ALS or Parkinsonism, and for this reason, the report is included with the VUSs as well. Next slide. Next case study. So this is a 16-year boy, 55-year female with hypertension and type 2 diabetes mellitus. There is a forgetfulness. She is known case of forgetfulness since last 3 years and shows slowness in walking. So with 3 years, since 3 years, and with visual hallucination. So all these phenotypes were used to add the phenotype-driven criteria to the analysis. These are the phenotypes which were added using the HPO terms available in Genomize. And the phenotypes driven ranking popped up a PACN1 gene, which is a missense variant and with the good coverage of 100x. The OMIM phenotype, OMIM disease assigned to this gene is autosomal dominant acne inversa familial, then autosomal dominant Alzheimer's disease, and then autosomal dominant cardiomyopathy, dilated cardiomyopathy or dementia, and frontotemporal dementia. So out of these phenotypes, the Alzheimer's disease is the one which was also suspected in the case with the family history, and hence this variant was reported. Also, there was the ACMG attributes given to this was PM1, PM2, PP3, and PP5, which is 2 moderate plus 2 supporting, classifying it as likely pathogenic variant, and hence this was reported as the variant of as a variant of choice in the report. Next slide, please. So there are multiple cases which we came across and helped us in analyzing the variant with basis of the phenotype driven ranking, and the classification was pretty good and straightforward here in the Genomize. So at Chromosome Labs, along with the whole exome sequencing, we also do the services for other services in clinical genomics domain, which are solid tumor panels. We have small solid tumor DNA panels. We have hereditary cancer panels, then comprehensive genome profiling with and high resolution HLA typing. In the research domain, we also offer services of whole transcriptome sequencing, whole methylome sequencing, long read sequencing on nanopore, along with the bioinformatics support. So with the whole exome sequencing, and the next slide, please. So with the genomics coming as a tool, it offers power to the knowledge what we have about the disease to take a precision medicine and to take a precision and to take a decision to the disease and to take a decision call on the disease phenotype and manage the patient. So for example, if the muscular dystrophies, if they were not treated earlier, if we just if we just depend on the phenotype, clinical phenotypes, and prolong the treatment interventions, it may result into multi-organ failures, as we see into iron overload diseases or in dystrophies where the conditions may worsen as the patient ages. So if we have clinical genomics handy tools available at disposal, we can definitely give justice and manage the patients very well uh, with the available knowledge and the precision medicines available.
Our next speaker is Dr. Kumar Gautam from Chromosome Labs. I'd like to introduce Dr. Kumar Gautam. He has done his PhD in genetics of renal disorders and has 14 years of experience in clinical genetics and molecular genetics. He has worked on more than 5,000 genetic cases, including rare disorders and cancer genomics. He has also worked on diverse forms of NGS-based data coming from different NGS platforms like Illumina, MGI, Nanopore, 454, and so on. Dr. Kumar Gautam is a senior bioinformatician in Chromosome Labs, and he'll be speaking on copy number variation analysis in practice.
So my topic is the analysis of CNV or CNV analysis in practice. So based on the size, there are three major classes of variants. One is SNV, that is one base substitution that may lead to amino acid changes. Another class of variant is indels, that is small scale addition or loss of nucleotides that may lead to frame shift. Usually it is less than 50 bases pair. Another class of variant is structural variant, that is large scale changes. Usually it's greater than 1 kb and it includes inversion and translocations. Copy number variants is a type of structural variants where section of genomes are altered and the number of repeat varies. Approximately 2/3 of our genome comprises of DNA repetitive regions, and among this, 4 to 10% of region comprises of copy number regions, which significantly contribute to the variation in the individual of our population. Next slide, please. Yeah. As said earlier, CNVs are the major part of a structure variant in the genome, in which number of copies of a particular genomic segment get duplicated or deleted. These copy number variants are technically ranging from 1000 bases to 5 megabases, and sometimes it's even larger than that. But most other CNVs are less than 10 kb of length. In this slide, you can see the first individual on the top has one pair of highlighted orange segment that is completely normal. In individual 2, at the left bottom, there are loss of one copy leading to heterozygous deletion. In individual 3, there is loss of both copies, that is called homozygous deletion. And in the fourth individual, there is a gain of one extra copy. And the fifth individual, there is a gain of 2 extra copies. Next slide, please. Yes. So the first question that comes in our mind is, what are the causes of these copy number changes and where exactly these things occurs. So the most common cause of copy number change are incorrect alignment of DNA during meiosis that leads to unequal crossing over. Other causes may include non-homologous end joining that happens while repairing double-stranded breakers without extensive homology. Environmental factors also plays a vital roles uh, that can cause CNVs. For example, agents that cause DNA damaging, such as ionizing radiation or chemical mutagens, that increases the frequency of double-strand break and subsequent copy number changes. Another not significant but major cause of CNVs may be attributed to transposons, like ALU elements are there, that can also trigger DNA damage and subsequent rearrangements. Talking about the occurrence, it can occur anywhere in the genome, irrespective of genic region or intergenic regions. It has also been found that CNVs are most commonly present in immunity or drug detoxification related genes. Next slide, please. Yes. So now there are different methods for identification of copy number changes. Earlier, it was detected using cytogenetic studies. One of the best example of such studies is karyotyping. Other method includes QCR quantification that to quantify specific DNA sequence related to a known reference gene reason to identify deletion or duplication. This is just quantification approach. Another method is MLPA, that is faster and highly accurate, but it is a targeted approach where it only identifies changes in the known region covered by the probe mix. Another approach is to use microarray CMAs. This is again high resolution method, and it can detect CNVs in multiple genomic regions in one go. It is cost effective, and as it is not targeted approach, there are possibility of finding and incidental findings also. Another approach is to use next generation sequencing machines, where we have whole exome sequencing, whole genome sequencings are also there, right? So using this approach, either whole genome sequencing panels or that covers multiple exonic regions can be used, or whole genome can be sequenced in one go. In WES, only coding regions are sequenced, so exact detection of breakpoints are only possible if the CNV breakpoint is present in coding region. In this approach, multiple exonic regions are scanned, so there are possibility of identifying incidental finding. Whereas in WGS based approach, whole genome sequencing based approach, all coding, all non-coding, intergenic, intrazenic, everything is covered. So here, exact breakpoint of CNV identification are easier compared to WES approach. In this approach, whole genome sequence, so huge data is generated, leading to high sequencing cost and time. Again, possibility of incidental finding cannot be ruled out over here because we are covering everything. We are sequencing everything over here. Next slide, please. Yes. So there are multiple implications that prompt us to study CNVs. In clinical genetics, it has enabled us to link DNA loss or gain with multiple neurodevelopment disorders like autism, intellectual disabilities, some disorders like neuropsychiatric conditions are also there, like schizophrenia is there, bipolar disorders are there, and also some of the complex rare disorders. Beyond neurodevelopment disorders, CNV are also known to cause cancer. As per recent studies, there are a lot of CNVs that are associated with increased risk of a specific cancer like HBOC, breast cancer. It is also found that certain CNVs can influence medication response in an individual and thereby impact treatment efficacy also. Understanding these CNVs can also help in predicting disease progression and patient outcomes. It also works as a tool to study population health and understand genetic diversity. As per few of the studies, high total CNV burden are strongly linked with increased risk of diseases. Targeting CNVs may also provide new therapeutic strategies for complex disorder, and CNV profiling can also help in personalized medicine approaches. So these are the implications. These are some of the implications that I found interesting of CNV studies. Next slide, please. So for calling CNVs, there are 4 approaches. These are read pair based approach, or read depth based approach, or assembly based approach, or split read approach. In uh, read depth approach, it is assumed that the number of read mapping to a region is proportionate to the copy number. It is effective for detection of both deletion and duplication, and common tools based on this is CNVnator or Contra. Another approach is read pair method approach. In this method, we compare the expected insert size of paired in reads to the actual mapped size. Discordant reads, such as those with large number of expected insert size, indicate deletion or duplication. Some of the best example based on this approach is breakdancer or PIMAS. Next approach is using a split read method. This method relies on analysis of reads that spans a breakpoint and only particularly mapping to the reference genome. This method provide high resolution, often base level detection of CNVs. And the last approach is assembly-based approach. Assembly-based approach. In this method, de novo assembly of the reads are done to construct a contig, and then differences are calculated by comparing it with the reference genome. There one more approach where tools where one more than two or more than two methods are applied together. This is done to increase the accuracy and sensitivity of the CNV calls. So the best example of this is Delhi or Manta. Next slide, please. Now let's discuss about the basic steps or framework of basic steps that are involved for CNV calling. As the most renowned approach is read depth approach, so I will be detailing about that part only. So yeah, it starts with mapping the reads, test sample reads to the reference genome, and then all QCs are checked, like mapping percentage, duplication levels, all those parameters are checked, and parallelly control samples are also sequenced and are also mapped to the same reference genome, and QC parameters are checked. And both of the alignments are then compared together to identify the differences in read depth. All such events where test sample read depth are highly disproportionate with the control samples are collected together in a VCF formatted file. These CNV calls are annotated with multiple layers of information coming from different databases. Some of these databases includes 1000 genome database, Nomad, ClinVar, etc. Once all informations are collated together, variant filtration based on clinical correlation are done to identify the reportable variants. Next slide, please. So this is analysis funnel. Yeah. So as per it starts with the analysis. Yeah, analysis of more than one. For any analysis or WES based analysis, at the start point there are more than 100 or 200 CNVs are there. Then multiple layers of filters like population frequency filter or removal of all known benign CNVs filter are applied. And at the end, genotype-phenotype correlations are done. And this way, more than 98% of CNVs are filtered out by this process, leaving just 0 or 1 to 3 reportable variant, depending upon the complexity of the cases. Next slide, please. So, there are benefits of NGS, but there are a few challenges also for CNV calling using NGS. So some of the challenges are written here. Like one of the most common challenges, sequence related bias. So in read depth based approach, it is highly sensitive to GC and uneven coverage. Therefore, normalization is done to distinguish true CNVs from the technical noise. If normalization of the control data is very good, in that case, it plays a very vital role in CNV calling. Next is repetitive regions. CNV detection is highly repetitive regions, like in ALU regions, is difficult because short read often map ambiguously to the multiple locations, leading to wrong estimation of read step, and consequently CNV artifact CNV calls. Next is small and rare event detections. For calling CNVs using WS approach, accuracy increases with the increase in number of exonic regions in the CNV calls. detecting a small single exon or low frequency somatic or mosaic CNVs is technically challenging. Often this leads to lower recall rate. Batch effect. Next is batch effect. Variable uh, batch effect. Variability in sample preparation and sequencing creates batch effect, making it difficult to distinguish genuine CNVs from the technical artifact, often requiring complex normalization. Some of the example of this that can be handled these things is exome depth and canons. Next is duplication detection. Duplications are generally harder to detect than deletions because they often cause smaller, hard to detect changes in the read depth. Next point is validation and standards. There is no single perfect method, and the relying on the short read sequencing often falls to match gold standard datasets that integrate long read and optical mapping. Next slide, please. Yeah. So this was all about how we call variants. So what are the different approaches for calling variants? So let's go for some of the interesting case studies that we have done till now. So this is one first case where this is a case of DMD. So DMD, BMD, EDMD was the differential diagnosis for this case. Here the patient was having proximal muscle weakness of both limbs since 5 years of age, and she was and he was having growth signs and contracture at the ankle and knees was there from past 2 years. Pseudo hypertrophy was also there, and bilateral muscle weakness was also there. And there was no family history, and this was a consanguineous case. And the patient age was 16, and male patient was there. So can you go to the next slide, please? For this case, we have identified a DMD mutation, and this is the genomic coordinate of the DMD, and it was approximately 131 kb. Homozygous deletion was there that extends from exon 2 to exon 5. And these are the coordinates, and we have reclassified it as pathogenic. And on the right side, these are the QC parameters, like what are the head-home ratio or TSTV ratios, and total number of SVs and small variants detected by our calling tool. Next slide, please. So this is another interesting case where 4-year male born out of consanguineous marriage with the case history was having AH. The case history of this patient, the differential diagnosis was having AHUS and TMA. Patient was continuous coughing, and hematuria was also present, and jaundice was also there in the patient from last 2 to 3 days. And there's no edema, shortening of breath was there, and urine protein to creatinine ratio was greater than 3.5. 7.25, yeah. It was elevated. So a differential diagnosis made by the clinician for this case was HUS and TMA. Can you please go to next slide, please? So for this case, we have done whole exome sequencing, and we haven't identified any single nucleotide change, but a huge heterozygous deletion was there in CFHR1 and CFHR2 gene. Both of these genes was deleted in this case. So and this was a very interesting case, and as we know that CFHR1 and CFHR2 are kind of disease causing polymorphism. This is disease-causing polymorphism, as it has evidence of more than 1% in Indian population. Next slide, please. This is another interesting case where patient was having differential diagnosis of nephrocalcinosis, and this was a 16 years old female. Consanguinity was not known, and the patient was having history of bilateral nephrocalcinosis as per ultrasound, and kidney size, both the kidney size was approximately normal, but medullary calcification was there, and multiple cysts was also identified. in the renal parenchyma. Urinary bladder was normally thick, and rest of the things are all histories are mentioned over here only, right? And talking about the family history, the patient's family has no history of any other renal abnormalities, but they have cardiac disease, and there was cardiac disease in mother and mother's family. Next slide, please. For this case, interestingly, we have identified a homozygous deletion in chromosome 20 that was approximately 30.81 kb, and that composes of CYP24A1 gene, and it extends from exon 5 to exon 11, and it includes 5-UTR region. We have classified this variant as pathogenic, and on the right side we have these are the QC parameters. For this case, we have generated approximately 9.52 GB of data. More than 95% Q30 data was there. Enrichment was also very good, above 82%, and PCR duplicates was around 10%, and mean coverage was nice over here, that is of 138x. Next slide, please. So till now, I have presented 3 cases that was related to CNVs, but this is a very interesting case where this is a case of SCA, where we have identified a tandem repeat expansion. So this was a case of SCA, and the patient was having difficulty walking, seizures, intention tumors, spasticity in lower limbs, hyperreflexia, and rest of phenotypes are mentioned over here, right? And this patient was male and 12 years of age. There was no family history, and the patient was born out of non-consanguineous marriage. Next slide, please. So for this case, we have done whole exome sequencing, and with the general CNV analysis, we haven't identified anything. We haven't identified anything in CNV, and we haven't identified anything in SNVs also. But after that, we have done repeat expansion analysis, where we have identified a heterozygous CAG repeat in PPR2A2B gene. And this gene was, as per that, in one copy there was 9 units of CAG was there, and in another copy there was 54 units of CAG repeats was there. And this gene was found to be related to SCA12, type 12. And after that, it was validated by MLPM. So talking about the repeat expansion, the normal range for CAG in this gene is approximately 4 to 32 CAG repeats. Intermediate is 33 to 50, and pathogenic is variant. This repeat expansion is called pathogenic in case if the repeat expansion is from 51 to 78. And on the right side, all these QC parameters like the total amount of data generated is 12 GB, and Q30 was above 93%. Enrichment was also very good of 84%, and mean coverage was very nice, 145x. So this has shown the strength of WES, that not only SNVs, CNVs, up to some extent, definitely there are challenges for detecting the tandem repeats, but up to some extent we can also detect tandem repeats using this technology.
Yeah. Thank you, Dr. Tolga, Dr. Heena, and Dr. Kumar for your time and taking it out, but also thank you to everyone who's attended. We hope the insights as a researcher, as a clinician, as a molecular diagnostic lab, you found the insights from the webinar useful. I think one key takeaway for me is there is an art and a science to reporting, and the art is which science to apply when. Genomize enables you with the science, whether you're a researcher, whether you're a clinician, the art is to use the correct science to apply it to your workflows. As Genique and Genomize, we are here to assist you, as is Chromosome Labs. If you have any questions, you can always reach out to us. We really appreciate the time that you've taken with us. It's been 2 hours, but quite an engaging session. But also thanks to Mandar and Shrishti, along with Muskan, who played a major role in arranging for this, along with the Genomize team and the Chromosome Labs team. We look forward to having more such sessions. And thank you for your time.
Thank you, everyone.
Yeah, thank you, everyone. Yeah.